What's up everyone, happy Tuesday. Welcome to a new series here on my YouTube channel where I will walk you guys through my master thesis this year, around maybe one video every week or 2 weeks I think. Today I have some lab work. I go to the lab around 3 days a week, Tuesday, Thursday and Friday. So I will take you guys with me. I'm in my 5th year of bioengineering, cell and gene biotechnology at the University of Ghent, but I'm doing my master thesis at the University of Antwerp. It's an awesome university. Make sure to check out their website and everything in the link below. My building, let's go. My day at the lab is over. It's basically 5 p.m. now, so it's 9 to 5 every time I have to go to the university for my thesis. And today I didn't really film anything because I didn't have that much work in the lab, so I did a lot of work on the computer in my office. And in the lab I basically did a bioscreen. This just means growing bacteria in different cultures and seeing which ones it grows the best in. So that's running now for 24 hours. For now, I'm going to the gym for an hour. Happy Wednesday, and today it's basically a whole day of this, following lectures and making notes. Alright, today I'm going to do a plasmid extraction to begin with in the morning. But I have a problem because my colonies haven't grown. That sucks. These are the colonies that should have produced the plasmid. Let's make a new colony by inoculation. Welcome in a gel electrophoresis room. I'm gonna put some DNA I already prepared on gel, meaning I'm going to figure out the size of the fragments of DNA inside this. It's from a restriction digestion, so I basically cut a circular piece of DNA in half, and now I'm actually going to check if that happened or not. I already prepared the gel. Let's now load this gel. You can clearly see the yellow stripes here. It's because you have to do everything with gloves because it's toxic because of the gel red in this gel. Minutes on the 120 volts. You can see little bubbles, so it's working. So what's happening, the DNA is going through the gel, and the bigger the pieces of DNA, the slower it goes through the gel, and the smaller pieces go very fast through the gel. Based on that, you can basically determine the size of each fragment when you compare it to the gene ruler. Alright, 40 minutes have passed, so we can turn this off. Someone else is using this one for the small one there. Then we have to take off this because we've used the buffer, and we're lucky because we don't have to change it. Let's open it up. Difficult with one hand. Now we have to put it under UV because gel red, the toxic compound, binds DNA and makes it fluoresce red under UV light. So that's here. Again, everything with the glove because gel red is toxic, carcinogenic. No bubbles. And this is to protect myself from the UV rays. And look at that. Isn't that nice? So based on this, I know the size of these fragments, and let's think for a bit. Yeah, it seems to be right. Yeah, I'm dealing with very big fragments, obviously. I used this reference here, so I know around my band is here, so it's about 5,000 base pairs long, for example. Awesome. Now I'm going to take those large, very flashy pieces because it all looks good. This normally should be my cut linearized plasmid, and I'm going to cut it out of the gel and purify the plasmids out of the gel basically. I have my scalpel to literally cut it out of the gel. The UV exposure time should be as short as possible because it damages the DNA. Here you can see my piece of agarose, which should contain all my linearized plasmids. I'm gonna put it in a little tube. And behold the two pieces of shell. Now I'm using this kit to clean up this shell and get the plasmids out of it. Let's go. Let's put it on 50 degrees to dissolve the shell. Meanwhile the autoclave is running because I'm sterilizing some media. Alright. It's just very hot inside under pressure, so it's 121 degrees. Alright, the autoclave has been running for 20 minutes on 121 degrees, 15 psi almost, so I can turn it off and let it cool down, and then my media inside will all be sterilized. These two are just inoculated with a bacteria culture from the -80. I'm going to incubate them on 37 degrees overnight. Alright, I'm back home in the plant cave. And to recapitulate what I've done today, I had a circular piece of DNA to begin with. I cut it open, I checked it if it was actually good, with the little bands, with the light, with the UV light. And then I put another fragment in between that plasmid. And with the Gibson assembly, which is just a collection of enzymes which put the plasmid inside and ligate it back together. So now in the 4-degree fridge I should have two little tubes with two new plasmids, each with a new fragment inside. Tomorrow I'm going to check if that's actually true by transforming the plasmid, which means just putting that little circular piece of DNA in a bacteria, letting the bacteria produce that plasmid, then check again on the little bands if it's the right plasmid with the fragment inside or not, because now I have no idea. But we will see that tomorrow. Well, actually Monday I will have the results, but tomorrow I will transform the bacteria. Going to do some Thai box. Alright, I have transformed the plasmids I made yesterday in E. coli host cells. So let's check it out. So the two new plasmids are now in E. coli bacteria. I did it with a heat shock via the warm water bath, doesn't really matter. And now I will grow these E. coli in the incubator for over the weekend, because it's Friday now. So these will grow, including the plasmids. They will produce the plasmids, and then on Monday, or no, on Tuesday, I will check if they actually contain the plasmid or not. In the meanwhile, I did a plasmid extraction of some other plasmids. Those are in the final steps. Happy weekend. I will use this Saturday to write on my thesis. This semester I will focus on a literature study about all the topics and background information about my topic, about my research area. It will probably take the whole day today because I want to write around 5 to 10 pages. So yeah, let's go. By now it's 10 p.m. still Saturday, and now I'm working on a video about the transgenic frogs.
The final day of the week, Sunday, I'm on my way to another training session of my sport. It's called Wing Revolution, by the way. It's like a self-defense system.
Alright, I'm back home. The training was very nice, and now let's feed the frogs. Watch my previous video if you don't know what I'm talking about. At the moment, I'm planning to do the frog experiment to make them transgenic, or at least embryos. Not this Friday, but next Friday I'm gonna start the experiment, so the 21st of October. But I don't have all the materials and equipment yet. But if it goes as planned, in the beginning of November, I'm planning to post the video of the transgenic frogs, which will be awesome. Subscribe if you don't want to miss that. I mean, fluorescent frogs. Who wouldn't want to see that, right? Sunday evening, I usually take the time to edit my videos if I have filmed any video that week or I still have to edit some video. And now I'm editing the video you're seeing right now, and I'm going to post it directly when it's done. It's almost ready, only this clip I still have to put in, of course. And then I just have to upload it on YouTube and do the keyword research, figure out the title and thumbnail, and make it available online. Thanks for watching, and I see you next time.